Okay, so chapter 4 is all about the central dogma of biology. So we have genes and cellular function. So if you remember the cell is divided into three separate compartments. Compartment number 1, compartment number 2, and compartment number 3. What are the three compartments that make up the cell? Structurally, anatomically, physiologically, functionally. What are the three compartments that? Excellent. So that's one of them. So the nucleus is one of the compartments of the cell. So the nucleus, remember we gave an example of the school. So the school has to have an administration, so that's the nucleus. What are the two other compartments that make up the cell?
The cell membrane.
Yes, so the cell membrane, the fence that gives the cell integrity and regulates who is allowed to pass through and who is allowed to exit and who is not allowed to enter the cell. So we have the cell membrane. And lastly, what do we have in the middle? Yes. Yeah, so we have the cytoplasm, and we said this cytoplasm resembles a swimming pool with rubber duckies floating on it. So there is a fluid compartment and there are rubber duckies floating on the swimming pool. So what are the two compartments that make up the cytoplasm? 1 and 2.
Cytoskeletal and organelles.
So we have the organelles, absolutely. That's excellent. So we have the organelles. So inside the school, if you're hungry, you go to the cafeteria. If you want to study, you go to the library. If there is a problem, you go to the public safety. So the same thing with the cell. If the cell wants energy, then there is the cafeteria would be the mitochondria. If there's any microbes invading the cell, the police would be the lysosomes. And we have the pita bread. What is the organelle that looks like pita bread? Stacked pita breads.
Golgi body.
Yes, the Golgi body. And the Golgi body is like the Amazon packaging. So the Golgi body is all about packaging proteins that are to be shipped out, exported outside of the cell. Because the cell doesn't just work internally. The cell also cooperates for homeostasis with nearby cells in the tissue and sometimes with very distant organs that are, we need to take the bloodstream. So the cell would be exocytosing and sending out some substance into the bloodstream to go to very far sites. So yes, the organelles. Cytoskeletal. So skeleton is like skeleton. So every cell has structural integrity and there's scaffolding proteins that make it look the way that it does. So cytoskeletal proteins are parts of the organelles that give the cell the integrity. But it is not the cytoskeleton. What is the fluid? The name of the fluid that's like this fluid in the swimming pool, and then the rubber duckies are the organelles floating on it. What's the name of it?
Cytosol.
The cytosol. Very good. So we have the cytosol. And we said the cytosol is what? Is it a solution? Is it a suspension? Is it a colloid? Is it an emulsion? It's a colloid. Excellent. And a colloid is a nice word for what? Colloid is, what does colloid mean? So apparently it's a fluid, but a fluid made up of what? We said a solution is a fluid that has really tiny particles. Yes?
It made up of protein and water.
Absolutely. So it's a fluid that is predominantly proteinaceous. Proteinaceous, and it's jello. It's like gel and white, colorless. Colloid. Okay, so now let's look at the administration inside the nucleus. You have to have an excellent understanding of the central dogma of biology. This is a very high yield concept that we all need to understand really well. The central dogma of biology. What is the central dogma of biology? The central dogma of biology, I'm going to erase this. The central dogma of biology is the track that enables us to obtain proteins. So we begin with the DNA. The DNA. Where is the DNA located? Where do we find the DNA? On the plasma membrane, on the cytoplasm, in the nucleus. Where is the DNA found? Yes?
In the nucleus?
Absolutely. So the DNA is found in the nucleus, and we perform a process known as transcription. So we transcribe the DNA, and transcription also takes place in the nucleus. So the DNA is in the nucleus. The transcription process, it's a process. Shin is a process. So there is a process we will talk about called transcription. So the process of transcription takes place in the nucleus and gives us RNA. Excellent. And there are 3 different types of RNA. RNA 1, RNA 2, RNA 3. What are the 3 different types of the RNA? So everybody give me one. Yes.
RNA?
Absolutely. So we have rRNA. And what does rRNA stand for those who don't know it? Yeah. rRNA stands for what? Yes.
Ribosome.
Absolutely. So this is ribosomal RNA. And this is the RNA that makes up the subunits of the ribosome. The 40S subunit and the 60S subunit and the 40S subunit are all made up of the ribosome. For eukaryotes, for bacteria are made up of 70S and 50S ribosomal RNA. And ribosomal RNA is so important. It is so important that it has a special place. There's a headquarters. There is a special place inside the nucleus that is specialized only for ribosomal RNA. What's the name of that special place inside the nucleus where we find ribosomal RNA? So remember, we have the nucleus. This is our nucleus. And inside the nucleus, there is a little tiny structure.
Nucleolus?
Absolutely, very good. So the ribosomal RNA is found inside the nucleolus. What's the other type of RNA? mRNA. And this stands for, mRNA stands for? Messenger RNA. And this is the transcript that gives us proteins. You know, proteins are important for life. What are the 2 different types of proteins out there? 1 and 2. Structural proteins. Excellent. What's an example of a structural protein? It's a protein that's there for structure, for construction, for structural tensile integrity. Keratin.
Microtubules.
Microtubules. Excellent. They have tubulin. Absolutely. Very good, Gardner. What else? Tubulin, flagellin, keratin, collagen. So those are proteins that are structural. They just stand there like staple. Cadherins, those are proteins that hook the cells together, attach them to one another. So we have structural proteins like keratin, collagen, and Barrington said tubulin, excellent. And there are functional proteins. Proteins that don't have a structure, but they have a job to do. What's a perfect example of a protein that goes around your body to perform a particular function? Yeah, functional protein. Give me an example of a functional protein. Functional protein. A protein that has a function, not just structure. It's not like staple that hooks up the cells together, like gap junctions, like tight junctions, or cytoskeletal proteins are example of structural protein. Now we need a protein that is not there for a structure, is there for a job to do.
Like the lysols. Like the lysols.
Like the what?
Lysols.
Lysols?
Lysosome?
Lysosome? So the lysosome is an organelle. It's not a protein. It is made up of multiple proteins, but it's not a protein. Yes, sure. Thiamine. So what is thiamine important for? Thiamine is an amino acid. It's like a vitamin supplement. How about hemoglobin? Hemoglobin picks up oxygen from the lung and delivers it to the tissue. And when the tissue metabolizes oxygen, hemoglobin takes oxygen from the lung, provides it to the tissue for oxidative phosphorylation, for aerobic respiration, and then metabolic waste is generated, carbon dioxide. Then hemoglobin would pick up the carbon dioxide and send it back to the lung. That's a perfect example of a functional protein, hemoglobin, HB. But what does the law say though? The law says you're never getting proteins unless you have an RNA. And you're never getting RNA unless you transcribe DNA. So the first step is you transcribe DNA in the nucleus. Then you would get RNA, mRNA. That's the one that we transcribe. And then we translate. That's another process. Translation. Another process is translation. Another process is translation. And translation does not take place in the nucleus. It takes place in the ribosomes. And obviously ribosomes are always found where? Sometimes in the rough endoplasmic reticulum, and sometimes free floating in the cytosol. And we will talk about why do we have two different types of ribosomes. What's the point? What's the significance? Why are there ribosomes on the rough endoplasmic reticulum? Why are there no ribosomes on the Golgi body? Why are there no ribosomes on the endosomes? Why are there no ribosomes on the lysosomes? Why is it gotta be only on the rough endoplasmic reticulum? So we will talk about that. But for now, that's the law. That's why they call it the dogma. Dogma means creed. The dogma is you start with DNA 1, you make RNA 2, and finally you get your target structural or functional proteins. Think about it this way. Can you go to the pharmacy and say to the pharmacist, I have strep throat. Can you please give me antibiotics?
No.
No. You have to go to the doctor. The doctor will give you a prescription. Then you take the prescription and you go to the pharmacy. So the doctor here would be the DNA. So the DNA is the clinic. We go in the nucleus, we transcribe the DNA. Then we get a prescription. But we don't call it prescription, we call it a transcript. because the process is transcription. Then we take that purple transcription and we go to the pharmacy. Who's the pharmacy? The ribosome. So the RNA is the transcript. RNA is the purple transcript. We take it to the ribosome, and the ribosome will read it and give everybody has a different transcript. Somebody wants a chemotherapy medication, somebody wants an antibiotic, somebody wants a vitamin. So it depends on what you have on your transcript. This is really, really, really important. Again, this is the central dogma of biology. It's a stepwise process to how to get target proteins. Target proteins. Let's say, let us say now, I'm living in the top of the mountain. There's not enough oxygen. So obviously, my body's gonna need more hemoglobin. Well, if I need more hemoglobin, I gotta go to the nucleus to get a transcript, RNA transcript that says hemoglobin. And then I go to the pharmacy, the ribosome, to read it. The pharmacy only reads. The ribosome only reads and it goes inside and gives you the medication you want. It is the DNA who dictates for us. And there are theories that says when Earth just started millions of years ago, there was originally RNA, and then DNA developed from RNA. So there was RNA first, then we have DNA, and then DNA developed or evolved from RNA. So is that clear? So we have DNA in the nucleus. It will undergo a transcription, and the transcription will give us an RNA transcript. And we said we have ribosomal RNA to make the ribosomes. We have messenger RNA to make the proteins, the target proteins, the target proteins of interest. And lastly, we have the tRNA. What does it stand for? Transfer. I wish that they call it translation RNA because that's what it does. But they call it transfer. Transfer RNA because it participates in the translation. Where does translation take place? It takes place in the ribosome. So who helps out in translation? tRNA. Who helps out in preparing the structures of the ribosomes? The rRNA. And what is the actual RNA transcript that we need to read in the ribosome and use the tRNA to bring the amino acids to make the protein polypeptide?
The mRNA?
That's the mRNA. So this is the central dogma of biology. Now when we go into the PowerPoints, we're going to take every step. We're going to take every single step by step. So we're going to talk about the DNA, then we're going to talk about transcription, then we're going to talk about the RNA and the 3 different types of the RNA, then we're going to talk about the translation, and lastly we will talk about the target proteins. But you need to have a GPS. You need to have a GPS of where the lecture is heading. Is that clear? Questions? So we're going to leave this and we're going to come back to it as we do the PowerPoint. Yes, Barrington?
Yeah, question. The 60S, 40S, 30S, what's that for again?
Okay, so the ribosome looks like the 2 buns of burger. So it has 2 subunits. S is a subunit. If you are a eukaryote, you have a 60S subunit and a 40S subunit. This is the ribosome of eukaryotes. For bacteria, known as prokaryotes, they have a 70S subunit and a 50S subunit. But in the end, everybody needs the ribosome, even bacteria. Bacteria that is known to not even have DNA, they have a small circular plasmid DNA, and they don't have a nucleus like us. They don't have organelles, but they gotta have a ribosome. Because if you don't have a ribosome, you don't have a pharmacy, you're not making any proteins. If you don't have proteins, you're not gonna have structure, you're not gonna have function. Structural proteins, functional proteins. Doesn't matter what protein are you will never have a protein unless you have a ribosome. Everybody gotta have a ribosome. What is the only organism that doesn't have a ribosome?
You say virus.
The virus, very good, because the virus relies on the host ribosome to translate its own DNA. So we will talk about those processes in more detail now. So let's go to the PowerPoint. Okay, so now we're going to begin with the first one, the DNA. What does DNA stand for? That's an abbreviation. So DNA stands for deoxy, and you need to know why they call it deoxy. Why do we call the DNA deoxy ribonucleic acid? So those are three words in one. It's a nucleic acid and it's a deoxyribose. So you see that the DNA is very long, like a football field. But how does it fit inside this little tiny nucleus? Because we have coils. We coil it and we tightly wrap the DNA threads around protein structures called the histones. And when you tie the DNA around the histones and you have the coil, it is now called the nucleosome. We will see that. So remember, no figure left behind. Every figure you see, you really need to master it, because the figures have all the key concepts for the lecture, for the lesson. Don't go past by the figures unless you really master them. This figure here shows you what? Even if I hide this, you should be able to look. Does this look like if I tell you what is this particular macromolecule, or what is this micromolecule? It can only be one of four. Either a lipid, either a carbohydrate, or a nucleotide, and lastly, or a protein. Does this here look like a protein? No, because we don't see an alpha carbon, we don't see an H terminal, we don't see a carboxylic terminal, we don't see an amino terminal, and we don't see an R substituent. So that does not look like a protein. Now, three are left. Does that look like a lipid? This look like a lipid. Do you see a long fatty chain, free fatty acids? Do you see a glycerol? No, so that is absolutely not a lipid. So two are left now. So this can either be a nucleotide or it could be a carbohydrate. Does this look like a carbohydrate?
No.
Be careful. Does this look like a carbohydrate?
Maybe.
There is a carbohydrate somewhere in there. Remember I told you when it comes to fructose and glucose, those monosaccharides, you don't need to know the structure, which one is fructose, which one is glucose, and which one is galactose, but you need to know what a ring monosaccharide looks like. What is that in the middle looks like? In the middle, in green. Is this a sugar?
No.
Yeah, that's a sugar. Remember the structure of sugars? So sugars are rings that have an oxygen bound to them. Remember I told you, you may not need to know that this particular monkey is a chimpanzee, that's an orangutan, but if you see a monkey, you should be able to say that that's a monkey. Maybe you don't need to know exactly which one is it. Maybe you don't know exactly what this sugar is, what this monosaccharide is, but you absolutely need to know that's a sugar. But the whole macromolecule is not a sugar. It has a sugar part of it, but it's not a sugar. Yes, Sheree.
So usually would phosphates always have the is that a double bond at the top?
Yeah, so we're getting to the phosphate. So you see now this is a macromolecule that is made up of three different micromolecules. You have something here in pink, green, and yellow. In green, I needed you to be able to recognize that green is sugar. Monosaccharide or disaccharide? That's a monosaccharide. One ring. Mono, one ring. Di, two rings. Oligo, up to 9 rings. Polysaccharide, more than 10 and above. So a nucleotide is so interesting that it has a sugar as a part of it. Sugar becomes a part of the nucleotide. In addition to the sugar in the middle, what is that in pink? Adenine. That's a nitrogenous base. That's a nitrogenous base. And lastly, you have a phosphate group. Those three, you need to know those three-headed beasts. The first head is the nitrogenous base. The second head is the monosaccharide ribose sugar. And the last head is the phosphate group. If you combine the three, you get a nucleotide. Yes?
Is it always a monosaccharide?
Absolutely.
Okay.
And a ribose at that
It's always a ribose.
is a ribose. For the RNA, it's called a ribose. For the DNA, is deoxyribose. D, what does D mean? D means devoid, minus. So usually, there's supposed to be an oxygen here.
Oh.
It's supposed to look like that.
Removing.
So when we remove that oxygen, this is really, really important. This oxygen here in the second prime carbon, we took away the oxygen. You see, the oxygen of this one stays,
Yeah.
but the oxygen of this one is gone. That's why we call it deoxyribose.
Like deoxidization.
Yeah, so deoxy, the removal of the oxygen. You need to know in yellow is a phosphate. So what is a nucleotide made up of? Number 1, nitrogenous base. Number 2, deoxyribose sugar. And number 3, a phosphate. That's a nucleotide. Yes.
Is it always shaped like this?
Yeah.
Okay. So then, like, if we were to see it under a microscope or whatever, it would be like that.
Yeah.
Okay.
This is the structure of a nucleotide. There's something you need to know. If I take away the phosphate, if I remove yellow, now I have only pink and green. That's it. What do we call that? Yes? Absolutely. Excellent, Sheree. Very good. So what's the difference between a nucleoside and a nucleotide? They're not the same. Nucleoside and a nucleotide. One with a T, one with an S. A nucleoside is only 2 out of 3. Only nitrogenous base, pink, and only glucose, green. But a nucleotide has all 3 and more phosphate. You could have adenine, deoxyribose, phosphate. How about ATP? ATP is adenine with the ribose and 3 phosphate, adenine triphosphate. You can remove the adenine, put guanine, now it's guanine triphosphate, cytosine triphosphate, CTP, GTP, ATP. Okay? So now this is a nucleotide. And we said that nucleotides are micromolecules. The DNA is the nucleic acid that is made up of multiple nucleotides. So we have 5 nitrogenous bases divided into 2 groups, purines and pyrimidines. Purines and pyrimidines. So the nucleotides, the nitrogenous bases are either purines or pyrimidines. You see, purines have 2 rings. 1, 2. 1, 2. Purines are big and they have 2 rings. They say pure or good. Pure or good. What does that mean? It means that purines are adenine, A for adenine, and G for guanine. Pure or good.
What did that say?
Here,
The second.
no, the second one is pyrimidines.
Oh, okay.
So pyrimidines are only one ring. It's very interesting. Pyrimidines have a really long name and only one ring. And purines have a short name but two rings. So the one with fewer letters is the bigger one with two rings. And the one with more letters is only one ring. So we have our purines, two ring, and our pyrimidines, one ring. You need to know something. Pyrimidines, in the DNA, you have a complementary base pairing. So adenine always binds with thymine, A and T, at. Where are you? At the school, AT. So adenine, thymine. Guanine binds with GC, GC, AT, GC. That's for DNA. So for the DNA, you have adenine binding with thymine and guanine binding with cytosine. Adenine binds with thymine with 2 hydrogen bonds, and guanine binds with cytosine with 3 hydrogen bonds. Is that clear?
Yeah.
If you look at the RNA, something special happens. The G still binds with 3 hydrogen bonds with the C, but the A binds with 2 hydrogen bonds, not with the T anymore. There is no thymine in your RNA. What replaces thymine in the RNA? Uracil. That is what the RNA looks like. That's the kind of questions on the exam. Who binds with who? With how many hydrogen bonds? A binds with T. How many hydrogen bonds? 2. G binds with T with 3 hydrogen bonds. And A binds with U with 2 hydrogen bonds. And RNA, G binds with C with 3 hydrogen bonds. So if you look at the, now let's look at the structure. Yeah, this is now gonna show us how they come apart. You see the DNA is double-stranded. The DNA is double-stranded. Double-stranded and anti-parallel. So the head, you know, the is like the husband and the wife, but they're mad. So the husband head is by the wife's feet, and the wife's feet are, yeah, that's how our parents used to do it back when they get mad. Now what do they do now? Sleep in a sofa, like Uncle Phil. Uncle Phil sleeping in a sofa. Yeah. But anyways, the DNA is double-stranded with 2 anti-parallel strands. So if you look now, there is a 5' to 3' end, and a 5' and a 3' to 5' end on the other side. Now you really need to understand this. What does the DNA look like? Double-stranded, anti-parallel, and double helix. So it's double-stranded, anti-parallel, and it's coiled around an axis, making loops like this. You need to know something very important also, that the bonds that make up one strand, you see, they're not dashed. Remember we talked about the difference between dashed lines and strong line. What does it mean when you see a dashed dotted line?
Broken up.
It means that it is a hydrogen bond, is a weak bond. That's why when you heat up the DNA, the 2 strands fall apart. When you cool it, they anneal, return back again. But the bond that is not dashed, the bond that is strong, black line, what kind of bond is that? That is an example of a non-polar what? Covalent. Covalent. That's a non-polar. Absolutely right. It's a non-polar covalent bond. Now, this non-polar covalent bond, if you heat the DNA up, is it gonna fall apart? No. Unless you heat it up like cremation. You know, like cremation temperature. Is it really hot? Yeah. Cremation. Yeah. I don't want to be cremated.
My mom does.
Really? Oh.
She
Yeah.
said
Cremation.
didn't throw me apart.
Cremation is hot enough to break up a covalent bond. But hydrogen bonds fall apart in much, much, much cooler temperatures compared to the covalent bond. Why? Because the bond is a hydrogen weak bond. It's not a strong covalent bond. And what we said earlier, the bond between G and C, how many hydrogen bonds are there?
3.
3. The bond between A and T, how many hydrogen bonds are there?
2.
2. The RNA, does the RNA look like that? Does the RNA have 2 strands? RNA. No, the RNA does not have 2 strands. It only has 1 strand. Okay. Alrighty, so what are the genes? What are genes? Genes are special regions in the DNA that code for a protein. So if you want hemoglobin, you gotta go to the hemoglobin gene to give you the hemoglobin mRNA to give you the hemoglobin protein. Yeah.
Would that be the same thing as far as like any like genetic mutations or like you have green eyes because your mom has green eyes and like
Okay,
stuff
we
like
gotta
that?
get to the mutations. We gotta get to the mutations. So let's take it step by step. I have DNA. That's my DNA, right? Double-stranded DNA. Double-stranded DNA. That's my double-stranded DNA. You need to know that this double-stranded DNA is divided into aisles. I like that. Aisles. Aisles. Genes are aisles inside the DNA, and each aisle has something. You know, when you go to the pharmacy, there's an aisle for antibiotics, there's an aisle for ointments, aisle for lotion. So the genes are aisles that when you go to them, they would code for something specific. So there is a gene for hemoglobin, a gene for keratin, a gene for collagen. So what you're saying is actually true. People with sickle cell anemia, there is a mutation in the gene for the protein that makes the red blood cell biconcave and discoid. So now the red blood cells become sickle. We will talk about that later. But now what we need to know is that the aisle, if you want to make a particular protein, you have to go to the DNA. The same if you have a respiratory disease, you go to the respiratory doctor. If you have a cardio disease, you go to the cardiologist. If you have a urinary problem, you go to the urologist. So same thing. You want a medication for a urinary disease, you go to the urologist. So you go to the right doctor. The DNA has separate places, separate aisles, separate genes. So if a cell now wants insulin, you had a meal, you had pancake. So right now your body is digesting, so it's going to be going to the aisles in the DNA where there is genes of insulin and transcribe mRNA for insulin, and then take the transcript of mRNA to the ribosome. Ribosome is going to read it, give you insulin. I'm not going to use insulin now because I'm fasting Ramadan, I haven't ate, so I'm not using that. I have other proteins though that are being demanded, the proteins that help me movement, proteins that help cognition, thinking. So everybody's metabolism is different depending on what you're doing. So you need to know what the gene is. Now you know what a gene is? Your DNA is not just a whole long linear chain. It is divided into regions. Each region is meaningful. It has a particular specific function. If something is wrong in that gene, you're never going to get the protein. You're never, that's why many diseases are out there for people like Ehlers-Danlos disease. There is a mutation where the proteins in the bony cartilage are not made, so they have really loose joints and they have problems walking.
Yeah.
Why? Is there something wrong with the ribosome? No, the ribosome says, give me the right mRNA, I'll give you the right protein. But you're not giving me the right mRNA because the gene is mutated. Because the gene is mutated.
So it's like you didn't give me the right thing, so I'm not giving you the right gene.
Absolutely, I'm not giving you the right protein.
I'm not giving you the right
You
protein.
didn't give me the right gene, I'm not giving you the right protein. So it's like you want to go and get a medication for asthma, but the doctor doesn't know it. So now you take it to the pharmacist says, hey, I have all the asthma medication inhalers back there, but you didn't give me the right prescription. So that's how the genetic diseases happen. It's because you damage the DNA, and then if you damage the DNA, you're never going to get the right mRNA. And if you don't have the right mRNA, the ribosome will never give you. The ribosome has no way of knowing what you need without giving it the right mRNA transfer. Are we clear? Okay. This is going to make more, as we keep going and going, things are going to start to become more clear. Remember, the DNA, is all of it active? Is all of it active? Do you know? Do you go to the pharmacy and take every single medication? No. So only some part of the DNA is active and the other part is lazy, off. Some part is on, the rest is off. Some part of the DNA is being transcribed heavily, like when you're having a meal, you're going to be transcribed proteins to help you digest, enzymes for digestion, chemical digestion. If you're not eating, if you're, let's say, reproducing, having reproduction, so you're going to be transcribing other genes. So the genes you're actively transcribing now, these are called euchromatin. 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 What is euchromatin? Euchromatin. Euchromatin is the on that is turned on. This is the on gene. While heterochromatin is the one that is off. You're not using it. So if you go to the pharmacy and you say, I want an ointment for my scar, for my burn, that's the euchromatin, what you need. You don't want no antibiotic, you don't want any medication for anything else. So all the other stuff you don't need is called hetero.
So that's like active and inactive, right?
Absolutely, active and inactive. The active part of the DNA, the genes that are active, that are being transcribed now to give me mRNA now, so I can translate it now, this is called the euchromatin. And the ones that are not being bothered, remember I told you about the coil? If I want some gene, I uncoil that part and I do euchromatin transcription. And the rest is so tightly coiled, sleeping off, that's called heterochromatin. Very good. Okay. That's the chromatin. We have Okay, we talked about this. You have the DNA, anti-parallel, double helix, and then you coil it around this coil. What's the name of the coil? The actual proteinaceous coil. What do we call that? Histones. Those are the histones. Now when you bind them together, when you bind the DNA around the histone, it has another name now. When the DNA is alone, we call it DNA. When the histones are empty, we call them histones. When you have now the DNA being tightly wrapped around the histones, what do we call that now? Nucleosome. So, what is we're going to look now, nucleosome. So you see now, the DNA winds around histone. You see where my mouse is? runs around histones to form core particles. Chromatin is gonna fold into a thicker zigzag fibers, and then in the dividing cell is gonna loop. The chromatin is looped to form thick chromatids, and then you end up with the X-shaped chromosome. How amazing is that? You have transcription factors that know exactly where is each gene in which nucleosome, and to uncoil it, unwrap it, expose it, and then perform the transcription to give us the translation and the proper protein. Yeah.
So is it a mutation when like for example, my son has blonde hair and green eyes, and I obviously do not.
Mhm.
So like, what does that mean? Because that's obviously in me that I like birthed him, but even my mom doesn't look like how does that work? Because if it's in you already and then you pass it on, but it's not on you, then it's a little.
Yeah, so you will see this when we do meiosis. You see when you do meiosis. So the haploid cell known as the sperm and the haploid cell known as the egg, two haploids come together to give you a diploid. It's amazing how some cells are diploid, some cells are haploid. Some cells have no DNA, no nucleic acid, like a bacteria cell. Everybody's a cell. Everybody has a bank account. Some people have $1 million in the bank account. Some people have $20, like me. Some people have $1 million. Some people have same thing. Some cells have a lot of chromosomes in them. Some cells have barely like threads, nucleic acid. But in the end, as long as you have it, you are a cell.
So it's like it's somewhere in there, and then just like
Absolutely.
depending on the other person, because their dad looks like me, like we're both brown, and then both our kids are like blonde and green eyes.
Yeah.
So it's
But
like,
up there in the lineage, in the process known as crossing over, and forming the chiasma, that leads to this diversity. So it happens.
So it's like in the little white.
Okay,
Yes,
adding to hers, this is physical. Can it also be, like, I play music and my kids, I notice, and it's kind of creepy, they can play music, like they're gonna play or they want to,
Like
they have
the
rhythm.
talent for
And
it.
then, their dad is like, nah, it's like in me. Is
You
this
mean like
personality wise?
yeah.
Like your
So the
talent,
personality
does it come
is
from that?
very complex.
Right,
The personality is an interplay
like the
of
talent, they can pass on.
your surrounding.
Right,
So where are you now? In which society? So your
Like I
children,
know
because
it.
the mom is the primary caregiver, you like the music,
like I
so
play.
now they're gonna look up to you and they're gonna do it. It's not that it was in their DNA to become musicians.
Because this is the thing I've noticed when they were little. I don't really do.
Yeah,
It's
so
like, I
it's
don't know. It's
yeah,
kinda
so
weird.
it's just something they pick up maybe from school,
Maybe yeah.
from you.
Like my dad does it.
Yeah.
I do it. They do it.
Yeah.
It's
So
weird.
it's not your DNA. It's not
Okay.
your DNA. Okay, so nucleosomes. Alrighty. You really need to know the structure of your chromosome. It looks like X shape. You see those red plates that are on the chromosome? See those red plates? Those red plates are known as the kinetochores. You see it? Kinetochores. Those are very important. Why are they important?
Together.
Because in cell division, the microtubules attach to them and then they pull apart and to separate them. Remember, we went over the cell division in mitosis. You need to have those kinetochores. What are kinetochores? Are they lipids? Are they sugars? Are they proteins? Are they nucleic acids? Proteins. They are proteins. They are the hook site because from the two opposite poles of the cell, the asters, centromeres, come out the microtubules, heading, targeting like magnet, targeting the kinetochores. Once they hook to the kinetochores, now they move in the opposite direction to pluck them off and separate them from each other. So now, ribonucleic acid. We talked about the different RNAs, and the bases are adenine, uracil, guanine, cytosine, and we have messenger RNA, ribosomal RNA, and transfer RNA. Now, genes, back to genes again. So somebody please describe to me what you learned today. What is a gene?
A gene is
Take a time, think about it, make a complete answer, and then tell me your answer. Don't make up the answer as you're going. Just take some time, ponder, contemplate, and then give me a full answer of what is your understanding today of what is a gene, because it really is a confusing concept. You see, it says a gene is difficult to define. In the past, they used to call it a gene is a segment of the DNA that carries the code for a particular protein. So every professor has their own way of explaining to the students what is a gene. But it's up to you now to, reflect what you have learned. So yes, please, let's begin with Natalie. What is a gene?
it's everything like your, all the information, right, from your DNA, and it passes on.
Okay, yes.
A
It has
gene
everything.
is the part of DNA that makes each individual an individual.
So I agree. The gene is a part of the DNA. Because the DNA is not only one big gene. It's multiple genes, little genes attached to each other. But how does a gene make you who you are? Yes, you had a hand up?
Oh no. But a gene, yeah. It's like a part of DNA that codes for a specific protein, and without it coding for that specific protein, your body's not gonna be able to produce all the proteins that you need.
Absolutely. Absolutely. Why do I like this answer? Because she went into the red zone. She said, if you don't have a gene, or if you have something, you will never get a protein. I like that. This is exactly the definition of a gene. It's the region of the DNA that gives you a protein, but if something is wrong in that gene, you're never getting that protein no matter what. If you give the pharmacist a purple transcript that doesn't have the medication, they will tell you go back to the doctor. So the ribosome is never going to give it to you no matter how much you beg. Yes.
So like if
It's not like when you beg with me. Can you extend the deadline? The ribosome is not going to beg. Yes.
Can you give us
So like if someone's short, like you're just gonna be short and that's it. Like if your gene is to be short, then you're short.
It's not your gene. It's the gene you inherited that was activated. That short person can have a tall. They can have, yeah. My two grandparents were tall. One of my uncles, the youngest one, is really short. I mean, really short.
Like,
And I had other, yeah, he's really short. And other, yeah. And my other grandparents, they were short. One of their kids was tall. You know, back in the day, they used to have a lot of kids. Like my mom has 8 siblings.
did I have to be?
Yeah.
No TV.
And some of them are tall.
What
Some,
day was it?
some of them are tall. Some of them are short. It happens. Okay.
Like, let me go back.
But hey, my grandparents are rich because all their kids are working and they're sending money. Their grandkids are sending money too.
That's why you have a lot of kids.
Yeah.
Yes, oh, yeah.
So that's one of the perks of having many kids. Okay, the genetic code. So now we're zooming in. We're zooming in. So we talk generally about what is a gene. So now we have to narrow down. So now we have to look exactly what does a gene look like. Okay. Okay, everybody wrote down because you have to upload this at the end of the lecture for your in-class credit. Okay. All right. So the genetic code is read in base triplets. So the DNA code is degenerate, meaning one amino acid can be coded by two different codons. So let's talk about that. So a codon is made up of 3 base sequences. So if I want to start transcription, I always begin, how do I know my gene? Because you know there's multiple genes, gene 1, gene 2, gene 3, gene for hemoglobin, gene for keratin. The process of transcribing the DNA is called what? What is the name of the process by which we transcribe DNA to get mRNA? Transcription, yes. Now, how does transcription begin? It begins with a start codon. You need to know what the start codon is. Absolutely, you need to know the start codon is called AUG. AUG. Everybody sees AUG? When RNA polymerase sees AUG, it knows that I have to begin transcription here. And the same way we have a start codon, we gotta have a end codon. We don't call it end codon, we call it stop codon. Stop codon. Stop codons are 3. You go away, UGA. You are away, UAA. You are gone, UAG. You really need to know those.
You are gone, you go away.
You go away, you are away, you are gone. UGA, UAA, and UGA. You go away, you are away, you are gone. You need to know what's the start codon?
AUG.
AUG. What are the 3 stop codon? UGA, UAA, UAG. You are gone. Very good, very good. Okay, now let us look at the. Okay, we really need to talk about this. So now let us. Okay, so let's talk about this. Is really important. Okay, so you have to know if I'm going from DNA to RNA, some enzyme gotta do that. So the enzyme is gonna give me a polymer because RNA is a polymer, isn't it? RNA is a polymer. And the enzyme is an ACE, so it will always be called polymerase. Polymerase. The enzyme that does the transcription of RNA from DNA is called a polymerase. The polymerase is going to give me what? Is going to give me what? The polymerase is going to give me RNA. So we call it an RNA polymerase. If you see something like that, it should make perfect sense to you. In the sense that RNA polymerase is an ACE, so it's an enzyme. Polymer, it means it's an enzyme that makes polymers of RNA. An enzyme, ACE means an enzyme. ACE means an enzyme. Polymer, it means, you don't remember polymers? Polymers of protein, polymers of carbohydrates. And RNA, that's our target. You also need to know what was it based on. So it was depending on DNA. So it's called DNA dependent. DNA dependent RNA polymerase. So now that should make sense to you. From the name, it's very obvious. It depends on DNA to give you mRNA. Is a DNA dependent RNA polymerase. DNA dependent RNA polymerase. So when you go from DNA to RNA, that's known as DNA dependent because you depended on the DNA to give you an RNA. Who's the enzyme that carries out the transcription of mRNA from DNA? What is it called?
tRNA?
What is the name of the enzyme that carries out the process of transcribing DNA to give us RNA? RNA polymerase, but DNA dependent RNA, because many kinds of RNA polymerases out there. So you have to be very specific. Is it DNA dependent RNA polymerase? Okay, in mitosis, remember we duplicated the DNA. If we have DNA, we make a copy and make another DNA. Remember I told you, if you have pharmacy number 1, you want to open up pharmacy number 2, you have to double all the medication, or you have to split the medication of pharmacy 1 in half and share it between 2 pharmacies. Listen to me, everybody listen to me. Let's say I have pharmacy number 1. I'm pharmacy 1. I want to open up pharmacy number 2 across the street. Do I split my medication in half and leave one half in pharmacy 1 and the other half in pharmacy 2?
No.
Or do I double every single medication that I have in pharmacy 1 and move it across the street to pharmacy 2?
Yeah, move everything.
So isn't that what happened in mitosis?
Yes.
In the S phase, remember the S phase in lab? We went over S phase.
Split into daughter cells.
So you split into daughter cells. Very good. So who did that though? What's the name of the enzyme who doubled, gave me 2 copies, 2X? Who gave me the DNA and made another copy of the DNA? It is also a ACE. So it's an enzyme. And it gave me polymers. So it's polymerase. What kind of polymerase is it? Excellent, because it gave me DNA, so it's the DNA polymerase. What did it depend on? DNA. So we call that DNA dependent DNA polymerase. Can you tell the difference between DNA dependent RNA polymerase, DNA dependent DNA polymerase? Is that clear? So the DNA dependent DNA polymerase doubles up the DNA in the S phase, of the cell cycle to prepare for mitosis. While DNA dependent RNA polymerase is going to be in transcription. And you have to know something about the HIV. In the HIV virus, it has something called reverse transcriptase. You see ACE, that means it's an enzyme. So how does it work? That enzyme has ability to reverse. What is the direction of central dogma of biology? From DNA to RNA to proteins. In DNA dependent RNA polymerase, we transcribe from DNA to RNA. In DNA dependent DNA polymerase, this is called replication. We replicate by duplicating the DNA, so we use a DNA dependent DNA polymerase. But the HIV, look at the arrow. Look at the arrow of HIV. In HIV, now who can help me name what is the enzyme? So obviously it's an enzyme, so it's an ACE. Does it make a polymer or no?
No. So like an A polymer.
It makes a polymer. Look at the arrow. The arrow is going from RNA to, so it does make a polymer. So it's a polymerase. What kind of polymer does it make? Yeah, it makes DNA. But what did it depend on? There you go. So it's a RNA dependent. It's an RNA dependent DNA dependent polymerase. This is a reverse transcriptase. So when the HIV virus hijacked your body, it will sandwich its RNA and then it will convert its own RNA back into DNA, and then hijack your ribosome to translate it. One second. Do you see? Do you see now that we have an RNA-dependent DNA polymerase? DNA-dependent RNA polymerase. DNA-dependent DNA polymerase. You got another difference between the three of them. Is that clear? Yeah.
So, like how you were saying before with the central dogma, if it started with RNA and then DNA came from RNA, then shouldn't that not be a disease? If that's essentially.
No. It's not a disease. This
Like-
happened millions of years ago when Earth just started. You know, there's many different theories about how Earth started. They have theories about how there used to be inorganic substance that later on gave rise to organic substance. You know, they say organic. What does organic mean? Organic. It has carbon and hydrogen. In the beginning of Earth, that's not how it was. It was all inorganic. Methane, and there was, before methane came, there was nitrogen, there was ammonia, and then they said millions of years passed, then the organic substance came. Same thing. DNA wasn't around. RNA was first.
Because
Then RNA developed into DNA. This here is the tactic that is used by the HIV virus. The HIV virus
So that's what it does.
doesn't have a
It's
DNA, so it uses a system, but it doesn't mean it's a disease. Going from RNA to DNA. It uses a system where the virus doesn't have DNA, it has RNA. But once it attacks your body, it's gonna now use a RNA-dependent DNA polymerase to reverse. That's why they call it reverse transcriptase, because it's gonna reverse, which is not unusual. This is a very wicked enzyme.
That's why I was.
It doesn't
confused.
exist, because usually our flow of information is in this direction, from DNA to RNA to protein. Only the HIV can go backward from RNA to DNA. So reverse transcriptase is a RNA-dependent DNA polymerase, which is unknown in biology except in this HIV. Okay, so now let's go into the PowerPoint. Let's go now into the PowerPoint. Okay. So now, if you look into the slide, we have transcription. So this is gonna take a while to come back. Okay. Transcription, not today. Transcription is when we're copying genetic instructions from the DNA to the mRNA, and that utilizes an enzyme called RNA polymerase. From the name, you can know what it's all about. RNA polymerase. ACE is an enzyme. Polymerase makes polymers for living. That's what it does. It's a polymerase. Oxidase causes oxidation. Reductase causes reduction. Polymerase makes polymers. What types of polymers? RNA. So utilize an enzyme called RNA polymerase, and like we said, with every C we add a G. With every G we add a C. T we add an A we add
U.
But remember, thymine doesn't exist on the RNA. So in order to get it on the RNA, we have A and U. Okay, you need to know this. So first we have our gene. You see the gene? Then we transcribe the gene and we get our nascent. What's the nascent mean? Nascent. Nascent. We have our nascent mRNA transcript. Here they call it pre-mRNA. Nascent means pre-mRNA. Meaning it's not ready yet. You know, when you take the cake out the oven, do you start eating it immediately, or do you need to chop up, do like a little surgery, remove like the side part and add whipped cream? That's exactly what we do. So the nascent mRNA is what just came out of transcription. If you look here, what I want is A, B, C, D, E, and F. But you see, there's brown parts that are extra parts that we don't need. We need to chop that out. We need to excise them, remove them. So if you look now, this is known as splicing, like a surgery. If you see now, we have A, B, C, B, D, E, A, E, and F. You see, they all have been aligned. The part that we cut out are called what? Introns. Why do they call them introns? Because they get to stay in the nucleus, they don't get to go out to the ribosome. Why do we make mRNA? Do we make mRNA so we can stay in the nucleus, or so we can leave and go to ribosome? So we can leave and go to ribosome. So introns get to stay in, locked in prison. Introns, they're not coming out the nucleus. They're not good enough. The ones who come out the nucleus and they get exported, that's why they call them exons. So they leave the nucleus and they go where? Into the ribosome for translation. You really need to know that. Introns get in, stuck in. Exons get to exit and they get translated. Yeah.
Can we say that it exits to the RNA?
It is all RNA. Even the intron is RNA.
Okay.
In fact, there's something called alternative splicing. What is alternative splicing? Alternative splicing. Alternative splicing. What is alternative splicing? Alternative splicing is all the chopped up introns that never got to make it out, all the introns that got to stay in, they get recycled. They get reassembled together into some useful type of exon that will later on be translated in the ribosome. You need to know what is alternative splicing. What is alternative splicing? Yes, please.
Leftover introns and extrons that are
Nope.
no.
Leftover introns that got to stay and get recycled later in the ribosome.
Absolutely.
Because they
Exons
stay in.
have exited already.
Okay.
Exons are what we needed. They have exited, they made it to ribosome, they got translated, they're done. It's those introns that keep piling up and piling up inside the nucleus, they're gonna cause defect. So that's why your nucleus is gonna recycle them and turn them into some useful exon and send them out to make something that is alternatively helpful to the cell. Yeah.
So if they try to recycle it and it's too much or it gets overwhelming, then that's essentially like a mutation?
No. If a mutation is when there is a damage to the actual DNA. This is mRNA. The introns are mRNA.
The messenger of the, like, communicating what the gene is.
Yeah. Yeah. So they don't get translated. Then you see after the splicing, we translate and we get our proteins of interest. Messenger RNA, we talked about that, and transfer RNA, ribosome. Okay, this is a transfer RNA. That's the hairpin structure. It's like a hairpin. The transfer RNA has three things. Number one, a hairpin body. Number two, an anticodon. And number three, the actual amino acid that we are bringing into the polypeptide chain. So, translation. Translation, we gotta watch a video. You can never get translation unless you actually watch a video of how the ribosome works. But before we watch the video, you need to know that the ribosome has three doors. Door number one, it has two subunits. We talked about that earlier, remember? Two subunits. It has three doors. 1, 2, 3. It has the exit door, E site. It has the P site, and it has the A site. So what happens is your mRNA is being transcribed and then it's being translated. So the first tRNA comes into the entry site. They don't call it the entry site. They gave it the letter A because it's the first one. So we enter through A, and then we shift into the P site, and then we shift into the exit site, but we deposit the amino acid into the A site. So let's watch the actual, this is something that you just cannot say. You have to watch it happen. You have to watch it. So let us watch. Okay, there's a really quick animation video that will help you understand. Okay, so let's look at this one. Everybody, please look. Do you see? How do I make this bigger?
If it's a short, I don't think you can. If it's a regular video, then.
Okay. Do you see the three sides? EPA?
Yeah.
A is entry. You saw the first amino acid, the first tRNA entered into the A site, then we shifted into the P site. Now look what's gonna happen. We moved the amino acid, then we shifted, then we exited through the E site. And then you see we transfer from P to A. That's how we make a polypeptide. Let's see if there is a better animation video.
Introducing Grammarly, like you've never seen before. Your all-in-one writing essential.
Steps of the translation process. Initiation on AUG. Has 3 sites.
You see?
The A site is the entry
The A site,
site for new tRNA charged with amino acid or amino acyl tRNA. The P site is occupied by peptidyl tRNA, the tRNA that carries the growing polypeptide chain. The E site is the exit site for the tRNA after it is done delivering the amino acid. The initiator tRNA is positioned in the P site. Elongation. A new tRNA carrying an amino acid enters the A site of the ribosome. On the ribosome, the anticodon of the incoming tRNA is matched against the mRNA codon positioned in the A site. During this proofreading, tRNA with incorrect anticodons are rejected and replaced by new tRNA that are again checked. When the right amino acyl tRNA enters the A site,
look, this is the mRNA. It has AUG. So the anticodon A binds with U. U binds with A. And G binds with C. And at the other end is the actual amino acid, methionine, phenylalanine.
site, a peptide bond is made between the 2 now adjacent amino acids. As the peptide bond is formed, the tRNA in the P site releases the amino acids onto the tRNA in the A site and becomes empty. At the same time, The ribosome moves one triplet forward on the mRNA. As a result, the empty tRNA is now in the E site, and the peptidyl tRNA is in the P site. The A site is now unoccupied and is ready to accept a new
You
tRNA.
see, it left, exited.
The cycle is repeated for each codon on the mRNA.
You see? And then we made a long linear chain.
Termination.
You see, you are gone, UAG. And then we stop.
Termination happens when one of the three stop codons is positioned in the A site. No tRNA can fit in the A site at that point, as there are no tRNA that match that sequence. Instead, these codons are recognized by a protein, a release factor. Binding of the release factor catalyzes the cleavage of the bond between the polypeptide and the tRNA. The polypeptide is released from the ribosome. The ribosome is disassociated into subunits and is ready for a new round of translation.
Okay, so you saw how it's impossible to actually just talk about, you have to see it. So now we saw that there are three different steps. There's initiation, so we start with the start codon AUG, and elongation we get to yes.
So essentially, like the messenger RNA was like the line that was at the bottom with like all the triple codes and stuff, and then the tRNA like moves across it like that, and then like that's how it kind of goes, and then makes a polypeptide, and then that makes us.
Yeah,
Okay.
that doesn't make us, it makes the protein.
Protein.
The protein. Then the protein is gonna fold upon itself. Primary structure, secondary structure, tertiary structure. Okay, so you must understand that one mRNA is being translated by multiple ribosomes. It's called the polyribosome structure. That's the polyribosomal structure. Okay, now we're back to what I was saying earlier. We have ribosomes in two different places. Where and where? Where do you always find ribosomes? In one of two places. No, there are no ribosomes in the Golgi body. In the cytoplasm, free floating, those are called free floating ribosomes. What are the other types of ribosomes? Ribosomes are either free floating on the cytosol, where else? In the rough endoplasmic reticulum. Why is that? Why do we have two types of ribosomes? One ribosome free floating and one ribosome on the endoplasmic reticulum. Yes?
Does the free floating one catch ones that are just
No?
looking for somewhere to go?
The free floating ribosomes, well, think about it this way. Whether you are a free floating ribosome or you are a ribosome that is bound to the rough endoplasmic reticulum, in the end of the day, you are a ribosome. And what do ribosomes make? They make proteins. So that's going to be the difference now. They both make proteins. Free floating ribosomes make proteins. Rough endoplasmic reticulum bound ribosomes make proteins. But the nature of that protein is going to be different.
Oh, one makes structural proteins. One
No. One, the cytoplasmic free floating ribosomes make proteins that are used by the cell itself. Because the cell itself needs proteins. But the rough endoplasmic reticulum bound ribosomes, they make proteins to be shipped out, to be sent out.
So the free floating make proteins.
That's for itself.
For the own cell, for the cell. You know how in a restaurant you have, yeah, I see your hand, just let me finish. You have in a restaurant food to be given to the customers outside, and food that the own chef eats and the waiters eat it.
Yeah.
So the cell itself, I don't know which one is better,
That's dumb one and stuff.
So you see
Like Chinese people.
the
Chinese
ribosome.
restaurant, they never eat their food, they making something else.
Oh wow. So the ribosomes make proteins to be used for their own self. Those are free floating. But the ones that are bound to the rough endoplasmic reticulum, they get a stamp, a barcode known as the signal peptide sequence. That barcode is going to be sent into the Peter bread. Who's the Peter bread organelle? The Golgi body. So the Golgi body is going to read, oh, this is signal peptide sequence. I got to package this protein into a vesicle so it can be shipped out. This may be going to the bloodstream. This may be going to the brain. This may be going to the heart. This may be going to the liver. So there's two types of proteins. Proteins to be used by the cell, and who makes those proteins? Free-floating ribosomes. Local, for local use. But if you're gonna ship it out, what are the ribosomes who translate proteins that are to be shipped out? Yeah. And they send them to who? To the Golgi body. Golgi body. You had a hand up?
Yeah, I was gonna ask, so this is the one where it kind of encapsulates it and then pushes it out of the cell.
Yeah.
Because I
This
remember
is-
from a couple of
this
slides
is-
ago.
This is the, if you see here, there we go. That's what I was talking about. That's the ribosome, and who is this? Don't tell me that's the Golgi body. That's the rough endoplasmic reticulum. So you see now, the ribosome would duck. It would duck its protein inside the rough endoplasmic reticulum, and the rough endoplasmic reticulum is gonna attach a signal peptide sequence, a barcode, and it's gonna send it out to who? To the Golgi body. We can see that here. That's the polyribosome, and you see that we went over this, how you have a start codon, and then the anticodon would come to give you the amino acids. There we go. That's what I was just talking about. Look at what's happening here. Blue is my rough endoplasmic reticulum. Green is the pita bread. So if you look now, the proteins that are coming from ribosomes on the rough endoplasmic reticulum are getting docked inside the rough endoplasmic reticulum. Then they come out and they enter into the Golgi body. Then the Golgi body, look what the Golgi body does. It sends it outside to be used to the outside of the body. You really need to know that. What's the significance between ribosomes that are free floating and ribosomes that are bound to rough endoplasmic reticulum? What's the difference?
Oh, the difference is ribosomes free-floating are proteins designated for the cell versus the ribosomes, I'm not saying that, RER. is
Yeah,
for-
rough endoplasmic reticulum. Well, RER is fine, but you need to know the rough. Why is it called rough endoplasmic reticulum?
Because it's texture.
Because it's ribosomes embedded on it, make it rough.
Oh, those are ribosomes?
Yeah, those are ribosomes. Remember, it's rough endoplasmic reticulum. Okay. So the same way we had alternative splicing and we had the post-transcriptional modification. When you chop up all the introns, that's post-transcriptional modification. But after translation, we have post-translational modification. We're always modifying. Modifying transcript, modifying translation. Okay, transport vesicles. Okay. Now, I like this figure a lot. Remember, no figure left behind. Every figure, you gotta sit down, look at it really well, what it's trying to tell you. Because it's trying to tell you a key concept you need for your understanding. You may skip a slide or two, but never skip any figure. All figures, you must know them. What does this figure tell me? Well, begin from large to small. I have obviously a cell. I can see that's very large. That's a very conspicuous cell. I see my Peter bread Golgi body. And I see on the surface of the cell a receptor. So the receptor is called prolactin receptor. Prolactin for lactation. I wonder what that makes, prolactin. Milk. So what happens is prolactin is a hormone. Prolactin is a hormone secreted by the pituitary gland, and then it binds to its like a lock and a key. Prolactin binds prolactin receptor. Testosterone binds testosterone receptor. Epinephrine binds epinephrine receptor. Nobody binds somebody else's receptor. So prolactin, ligand, stimulus, hormone binds prolactin receptor, and then that leads to an intracellular signal transduction cascade, a stepwise sequela of events.
What?
Okay, listen, just listen. You have to listen till I finish.
Yes.
You have to let me
That would
finish.
be a lot.
Don't raise your hand. Don't interrupt. Just listen till I finish. It's gonna make sense. Just give me a chance till I finish all the way to the end. I have a prolactin. Is gonna bind into the prolactin receptor, and once it binds, there's gonna be an intracellular, meaning inside the cell, signal, meaning there is multiple signal, transduction, meaning we're transducing, one is giving the other, like a dominoes effect, like exactly, signal transduction, cascade is like a line of events. There's like a sequela, one after the other. And if you see here, that's going to lead to ATP being converted into ADP. When ATP became ADP, that means I lost a phosphate or I gained a phosphate. I lost a phosphate. This phosphate that I lost did not go in vain. You see it? This yellow phosphate, it went and it bounded this purple regulatory protein. Once it bound to the purple regulatory protein, it kick-started it. It activated it. It turned it on. The regulatory protein now started to move, mobilized, it entered into the nucleus, then it caused transcription. You see, the RNA polymerase with this regulatory protein is transcribing which gene? The casein gene. The casein gene is the gene for milk. Then once we transcribe, we have an mRNA. The mRNA was translated where? Who is this in blue?
Rough endoplasmic reticulum.
Rough endoplasmic reticulum, ribosomes. The rough endoplasmic reticulum does not translate. The rough endoplasmic reticulum ribosomes translate. And now you know this is going where? To the Golgi body. The Golgi body took it and then packaged it and then emptied it out into in the milk so the baby can drink it. So it's looked like a long sequence, is not. It's very simple. It's trying to tell you that I get a hormone from the outside that makes me on the inside as a cell undergo transcription, what we just went over. Then I hand over the mRNA transcript to the rough endoplasmic reticulum to put a barcode, signal peptide sequence on it. Then I send it to the Golgi body. Then I'm gonna release it into the outside of the cell to mix it with the colostrum and mix it with the milk duct, and then the baby is gonna suckle the nipple, and then the milk is gonna come out. Yeah.
So essentially, like, you know how like the mom's breast starts to hurt even when the baby's crying? Some like they could be crying in another room and your boob will start to hurt. So essentially that's what's happening in that like quick second of time. while that process is
Because
happening.
it's a cognitive thing. Because when your mind, so this is that's a very good observation. Because
It is a fact.
sometimes when the baby suckling the milk, the nipple, there is an irritation of the nerve endings on the nipple. This irritation would irritate the mechanoreceptors that would signal action potentials that would go to the brain in the hypothalamus that cause a positive feedback release of the milk for the baby. Also, what you mentioned, you as a mom thinking, maybe the baby didn't start suckling the milk yet,
It could be in the other room.
but your brain, Your brain knows the baby woke up, the baby wants milk. That causes the premature release of the. That's something interesting, actually.
Yeah.
It's
Let
very
me ask
weird.
you, some women, they can't produce as much. That's a problem because of the transcription, right?
Some women they cannot produce more milk.
Yeah,
some can't produce
because
at
I
all.
read a lot. Some women,
Yeah, I don't know. That's interesting, but I don't know why.
it's right here.
Do you know why, Dr. Allen?
What was the question?
The question was, why is it that some women have produced a lot of milk, some women are lacking, like unable to produce enough milk?
It could be due to the hormonal levels in the blood.
Yeah, it's
So
insufficient.
the hormonal levels can vary, and that could lead to lesser production or greater production.
Yeah, it's the hormones. And what makes the hormone levels drop for the milk? Like the prolactin levels go down. Maybe it's like a pituitary problem for some females. Oh. So it's the hormone. The hormone is not around. This hormone up there, prolactin. This is the one that makes the.
lack of proactive.
So from the name prolactin production of milk. Yeah.
So it says like not breastfeeding or pumping milk and like stop and signal the brain, okay, I need to stop. I don't hear, I don't feel baby no more. So that's why it stopped. You know?
Yeah.
When you're away from your baby, that's why they want your baby close to you right away.
Right.
Like
Yeah.
when you give birth.
And so when you don't use it, you lose it.
Right.
If the baby is not suckling the milk, the milk stops coming.
But prolactin is not the only hormone involved. So you have oxytocin.
Yeah.
You also have progesterone for
The
the
thyroid.
rest of the mammary gland.
Yeah.
So you must have the proper balance of all those three hormones.
Yeah.
Yeah, so like if you're
Progesterone,
depressed, you
oxytocin,
can
prolactin.
Yeah, listen to the end. Yeah. So prolactin is for milk production.
Yeah.
Oxytocin is for milk exit, so that milk can come out the milk injection, and progesterone is to stimulate the process of the mammary gland secreting the milk. Thank you, Dr. Allen. Okay, so now let's move into, that's another figure I like too. You have to be friends with these figures. No figure left behind. You have to know your figures. So if you look now at this one, who can interpret? You have to have the skill of interpreting the figures. So you see now, we have luteinizing hormone. Luteinizing hormone is a hormone that for males, it gives testosterone. For females, it makes estrogen. It's amazing how you have an apple tree and a carrot tree, and both of them you give water, and then they give you fruits. Even though they're two different, apple, tomato, green peppers, carrots, watermelon, they all grow with water. Same thing, luteinizing hormone. So you put water in strawberry, gives you strawberry fruit. You put water in banana, gives you banana fruit. Same thing, luteinizing hormone. You put it to a female ovary, will give you estrogen. You give it to a male testicle, will give you testosterone. It's the same hormone, will give you two different effects. So now let's look at what's happening here. You have a luteinizing hormone is gonna, you see, have a signal transduction cascade, and this signal transduction cascade is going to, so we have a premature mRNA. The mRNA is out, translated. Now we have an enzyme. This enzyme is laying in there. The enzyme is laying in there. So the second messenger is going to kickstart the enzyme, and it's going to convert for us cholesterol into testosterone. So this figure is teaching you that testosterone is made up of what? Cholesterol. Okay, when it comes to DNA replication, we talked about that, and we said that DNA replication, we use what kind of enzyme? DNA polymerase, DNA dependent DNA polymerase. And when we look at this figure here, DNA replication is a process that is known to be semi-conservative and discontinuous. What does semi-conservative and discontinuous mean? Semi-conservative means We start off with a parental strand, and then we make a copy to get a daughter strand. That's why it's semi-conservative. Start with a parent, you end up with a daughter. Semi-conservative, not fully conservative. Semi-conservative, partially conservative. Discontinuous because they are anti-parallel. There are anti-parallel strands, and then we're unable to perform the replication in one direction. So in one direction is 5' to 3', because the other one is inverted upside down. It's going to be 3' to 5'. So the process of DNA replication is going to be very discontinuous, because you're going to need this blue enzyme called DNA ligase. DNA ligase is going to come like glue, is going to be connecting all the missing discontinuous pieces to make them whole again. You need to know the DNA helicase. That's the one that unzip and unwind the double-stranded DNA. You're going to unzip it, unwind it. Okay. All right. So the DNA damage response is the DNA polymer occasionally, rarely makes mistakes. So there's always DNA damage response. If there was any type of damage that happens, there are mechanisms in place to correct the replication errors. So in the mutation, you have changes in the DNA structure due to replication errors or environmental factors. Now, when we look at the cell cycle, we discussed that. We said that the cell cycle has two main phases. Interphase, that covers G1 phase, which is the phase for growth and normal metabolic processes. Synthesis phase is the DNA replication. And second gap phase is growth and preparation for mitosis. And then you have those mitosis. And mitosis, you have prophase, metaphase, anaphase, telophase, and cytokinesis. If you look now, there's a first gap phase, so it's an interval between the cell division and DNA replication. So the cell carries out normal tasks and synthesizes proteins and materials for the next phase. S phase is when we replicate the DNA, and the G2 phase is the second gap phase. Okay. So you need to know the distinctive structures of mitosis. Prophase is when the membrane of the nucleus fragments and disappears. So you see here in prophase, this is prophase. You have the nuclear membrane breaking, so the chromosomes are going to condense and the nuclear envelope breaks down, and the spindle fibers grow from the centrioles. See, these are the centrioles. Have the spindle fibers going to go and target. Remember, we talked about the kinetochores. So they are going to target the kinetochores so they can pluck them off. And we have the metaphase. In metaphase, you have the chromosomes lined up in the middle in what's known as the metaphase plate. When all the chromosomes are lined up in the middle, the metaphase plate, so you have chromosomes lie along the midline of the cell, and some spindle fibers attached to kinetochores, and you have the fibers of the ester attached to the plasma membrane. In anaphase, the centromeres divide into spindle fibers, and they pull the sister chromatids to opposite poles of each side. And then you have the cleavage furrow starting to arise, and that's what's going to cause the two cells to separate. In the karyotype, so heredity is the transmission of genetic characteristics from parent to the offspring. And the karyotype is a chart of all 46 chromosomes that are laid out in order by the size. Sometimes you have homologous chromosomes. Homologous chromosomes, we have one chromosome from each pair, inherited from each parent. So homologous chromosome, meaning you have half of it from each part, from each parent. There are 22 pairs of autosomes that look alike and they carry the same genes. There is one pair of sex chromosomes, one pair of sex chromosomes, and those are the X and the Y. So the sperm has an X chromosome and the Y has a Y chromosome. That's the karyotype, and it shows you all 22 have two pairs, but if you have an X and an X, that makes you a female, and X and a Y, that makes you a male. So you have diploid, we talked about that earlier, diploid cells. They describe any cell with 23 pairs of chromosomes. You have haploid cells, these describe cells that contain half as many chromosomes as the somatic cells. The germ cells, those are the sex cells, the eggs and the sperms. The eggs and the sperm, and they include the sperm and the eggs, and they're haploid. So the fertilization is going to restore the diploid 2n number to fertilize egg, and the somatic cells are going to arise from it. Gene locations and variants. You have the locus, which is a gene's position on the chromosome, and the alleles, you have a combination. You have a dominant allele and a recessive allele. If you have two dominant alleles, that makes you homozygous dominant. So you have the dominant trait. If you have two homozygous recessive, two small letters, that makes you homozygous recessive. If you have a mix, one dominant, one recessive, that makes you still dominant, but it's known as heterozygous dominant because it's a mixture. If you look at sickle cell disease, you see the problem is you have homozygous recessive sickle cells because there's a genetic mutation. We're unable to make the proteins that help ensure that the red blood cell is biconcave and discoid. So you see that people that have homozygous dominant have healthy shape, heterozygous dominant have a healthy shape, is the homozygous recessive that looks sickle. So the parents can be healthy, heterozygous carriers of hereditary diseases. Genetic counselors, they perform genetic testing and advise couples on the possibility of transmitting genetic diseases. Multiple alleles, codominance and incomplete dominance. We're going to talk more about that on the circulatory system. So polygenetic or multiple gene inheritance, we have genes at two or more loci. So you have the eye color, the skin color, and some forms of cancer. And pleiotropy is when one gene produces multiple different phenotypic effects. And that's in a disease known as alkaptonuria. In alkaptonuria, you have a mutation of the gene on chromosome 3 that results in a darkening of the skin, sclera, and the urine. Sex-linked traits are traits that are passed only by the germ cells, by the X chromosome and the Y chromosome that are inherited from the sperm and from the egg. So those are the sex-linked traits. If you look at the, yes?
So I actually had a question about this, but I was waiting for it to come. So I have, I think, O negative blood, and then my son has O positive blood, and then when I had my daughter, well, after I had my son, I had to take the Rogram shot.
Yeah.
Because if I had another child, they could die because we're both O.
Absolutely.
blood.
No, it's not because you're O negative and he's O positive. If he was O negative, you wouldn't. But the Rogram shot is what saved the child. So yes.
Is that the same shot that you take while you're pregnant? Like, cause like for example, my mom, I think she's O negative, and she was pregnant with my brother, and he was like, I think A or B or something like that, and they had to give her a shot so that her body wouldn't attack him.
Yeah.
So let's go over that.
Yeah, I used to take them to a pregnant woman. Pick it up from the mat and take it to labor and delivery.
Take the shot on and fake it and become a big.
For real. You're less negative.
Yeah, I'm negative and then
He gives blood to everybody.
and then I can receive from anybody, but I can only give to him, and then he can give to anybody, but only receive from me. And then I think my daughter's 80.
Okay, let's go over that. That's very important, actually.
I had to double check that.
That's cool.
Okay. You must first understand what does antigen mean. So an antigen is always on the surface. You know, the red blood cell is like that. If you are on the surface of the red blood cell, you are an antigen. Some red blood cells have A antigen on the surface. Some red blood cells have B antigen on the surface. Some red blood cells have A and B on the surface. And some cells have nothing. They are bare, and there is nothing on their surface. Is that clear?
Yes.
Is everybody following? Those are red blood cells. If you are on the surface, that makes you an antigen. This is A antigen. That's the A antigen. That's the B antigen. That's the A antigen. And that's the B antigen. Does O have any antigen?
No.
It has no antigen. So the law says you have antibodies against all antigens that you don't have. So, what are antibodies? Antibodies are weapons. Antibodies are weapons. So A antibodies, that means if you have A antigen, according to the law, you will always have antibodies against all antigens that you don't have. So somebody with an A antigen, they're going to have antibodies against what?
B.
Against B, because they don't have B. So somebody with A, they will have anti-B antibodies. That's true. Isn't that true?
Yeah.
Somebody with A antigen will have anti-B antibodies.
And that's your blood.
Your blood type is A.
Right, I'm A.
So you will have anti-B antibody. Because you're A, you're going to kill B.
Right.
You're not going to have anti-A. You're A and you have anti-A. You're going to end up killing yourself.
Right.
Does it make sense? So everybody has antibodies against what you don't have. So anti-A will have anti-B. What about B blood?
A.
They will have what type of antibodies?
A antibodies.
They will have anti-A antibodies. They will have anti-A antibodies. They have anti-B, so anti-A antibodies. Anti-A antibodies. Are you clear? What about anti-A and what about antigen A and B? Both. You have plus both of them. A and B. What does that mean? Yes.
Do they fight the O antibodies, or do they not fight?
What
the
antibodies will A, B antigens have? Raise your hand if you know the answer.
B,
Yes.
B antibodies.
If they would have B antibodies, they will kill themselves because they have B too.
O? They don't have?
They don't have any antibodies against nobody because they don't have against A because they have A on them.
And
They don't have anti-B because they have B on them.
so they can receive from everybody.
And they will never have anti-O because O means nothing. You don't have anti-nothing. You don't have anti-nothing. You gotta have anti-something on a surface. This one has nothing on the surface. So you can't have anti-nothing. Anti-O. O means nothing. So this one, you said something very nice.
It can receive from all of them.
It can receive from all of them. It can receive from A because it has no anti-A antibodies. It can receive from B because it has no anti-B's antibodies. And it can receive from O because nobody has anti-O antibody.
What's your type?
Yes,
So when you have A and B positive, what do you mean?
we get into that. Don't jump the gun. We get into that. So now let's stay in this. Let's stay in this. So we have A and B. You have no antibodies against nothing. You are universal.
Don't tell her.
You're a universal recipient. Because if they give you blood type A, you have no anti-A antibodies. They give you blood type B, you have no anti-B antibodies. They give you blood type AB, you have no anti-AB. They give you blood type O, you have no anti-nothing. So don't see this plus here confuse you. That's a different thing. I'm just trying to tell you that it's A, B, you have both. But it doesn't mean positive. We get into positive. It's not there. We're not there yet. And lastly, O. What does O have?
No antibodies.
O has
No antibodies.
O has no antibodies.
No antigen.
O has no antigens. The law says you will always have antibodies against
Antigens.
that you don't have. So what antibody does O have?
A and B.
A and B, because it has no A, so it will have anti-A, antibodies. And it has no B on it, so it will have anti-B antibodies. So AB, so that means O can never receive blood except from O. But everybody can accept O because nobody has anti-O. O has anti-everything because O has nothing on its surface. O has anti-A because it has no A. O has anti-B because it has no B. O has anti-AB because it has no A or B. Do you get it?
Yes.
Now let's move to what Barrington was talking about, the plus convention. The plus is about the mom. You have a mom who's pregnant with a baby. That's the baby. If the mom, this is a new convention called the RH, the rhesus factor. The law says if the mom has RH on the surface, so the mom can either have RH or be bare, no RH. If the mom has a RH factor, what kind of antibody does it have?
A and B.
No. A and B is a separate thing. We're done with A and B now. It's new. It will have no antibodies. But a mom that has no RH, it will have what? Antibodies against what? You with the guys with the AB, forget about AB is over. AB is done. We're talking about RH. You either have anti-RH or you don't have anti-RH. Does RH have anti-RH? No anti-RH, because if I have anti-RH antibodies, I'm gonna kill my own self. But this one doesn't have RH on a surface.
No.
So that means it will have,
RH antibodies.
yeah, and we call them anti-RH. We don't call them RH antibodies. We call them anti-RH antibodies. And because they're anti-RH antibodies. You have anti-RH antibodies. Yes.
Does that have something to do with RH null? Like
RH what?
RH null? Like, cause isn't there like a rare blood type like that it doesn't have RH in the blood or something like that?
Let's stick to this. Let's stick to here. Let's stick to RH and get past this point. I have two types of RHs. I either have RH, I'm RH positive, or I have no RH. If I'm RH positive, do I have any antibodies? No. If I'm RH negative, meaning I have no RH, that means I will have anti-RH. Now let's look at the mom. If the mom is RH positive and the baby is RH positive, is the mom going to have antibodies against the baby? Is the mom going to have RH? Is the mom going to have antibodies against the baby? No, because you're RH, I'm RH. We're compatible. If the mom is RH positive and the baby has no RH, will the mom destroy the blood of the baby? What does RH minus mean? RH minus means you have nothing. RH means you have antigens against the RH, but you have no RH on you. The baby doesn't attack the mom. The one who attacks is the mom.
Yeah.
If you're RH positive and the baby is RH positive, it's all safe. If the mom is RH positive and the baby is RH negative, is the mom going to attack the baby? No, because the baby is like, oh, it has no RH on the surface. You always have antibodies against what you don't have. The mom has RH. It doesn't have any antibodies now.
So she has antigens of negative RH?
No, she has RH. There is nothing called negative RH because negative RH means it's empty on the outside. There's no RH on the outside. There is no RH on the outside. So if the mom is RH positive, she's not going to attack the RH negative because there's nothing to attack. It's empty. It's RH minus. It's bare. There is nothing on the outside. But if there was something on the outside, RH positive, it's still not going to kill it because the mom herself is RH positive. So here, RH positive is okay. RH positive, RH negative, there is no RH, there's no RH, still okay because there's nothing to attack. Now let's look at when the mom is RH negative. So that means she has what type of antibodies? She's RH negative.
Anti-RH antibodies.
Absolutely. The mom is RH negative. That means cannot tolerate any RH minus. So now let's say the baby is also RH minus, meaning it has no RH on the surface. Will the mom kill the baby?
No.
No. Because we both don't have RHs.
It's compatible.
It's compatible. The issue is when the mom is RH negative and she has anti-RH positive antibodies and the baby has RH positive on the surface. Then they're not gonna match. So what's gonna happen is on a first pregnancy, you see, God is very merciful. On a first pregnancy, the baby is gonna live. But the antibodies are ready and angry and waiting for the next pregnancy. If the next pregnancy, another RH positive baby comes, then the antibodies that were made in the first pregnancy are gonna kill the baby. Hold on. They're all gonna kill the baby. Is that clear?
Yeah.
When the RH minus and there's RH plus, that's when the baby gets killed. So when we now say A positive, it means is blood type A and Rheus is positive. B positive, it means is blood type B and Rheus is positive. Blood type AB negative, meaning it's blood type AB and its Rheus is negative. These are two different conventions. They're not the same. The letter is for something and the negative is for RH. So it should say this. A negative is actually A RH negative. That's what they mean. B positive, it actually means B RH positive.
Yeah.
Okay, yeah.
This is why, like, back in the day when you said people had a lot of kids and, you know, there was no TV,
Mm-hmm.
it was not technology. So this is why the firstborn would live. Then when they would have more kids, they would die. Like, their kids, they don't know why, they'll just die. You know what I mean?
Like those people have like
Sonograms.
16
Yeah.
Yeah, I don't know why they died, but
It could be a lot of stuff.
yeah, many things.
It could, that's what I'm trying to say. Like, it could have been.
No, so that's why you reminded me. Every RH minus mom, we give her a shot of the row gam. Are you following me?
Yeah.
We give her a shot of row gam, and the row gam is gonna make the mom RH positive. So now, forever, when you give the mom RH row gam, she's gonna be RH positive forever. So that means she will never have. Let me finish. If the mom is RH positive,
Right.
if the mom is RH positive, so that means she will never have antibodies against RH. So if the baby is RH positive, so am I, because I got the ROGAM shot.
Right.
If the baby is RH negative, it has nothing on the surface, we don't target that. Yeah.
Okay, I'm RH positive, she's RH negative, but they gave her the shot.
Yeah.
The second dose.
So now
she's
RH
RH positive, but her blood type is O negative. I'm
Or
A
does it-
positive.
does it
change?
Yeah, so she's O positive now. The O doesn't change. The O doesn't change.
Positive.
The RH, the Rheus changes, yeah.
Welcome to my side.
Yeah. Okay? All right. Okay, so now let's finish with okay, this shows you see the transcription again, making the mRNA, transcription, and this mRNA is gonna give you enzyme 1, it's gonna convert phenylalanine to tyrosine, and then you're gonna convert tyrosine into melanin. If one of the enzymes is defected, you cannot move over. If enzyme 1 is not working, you will never get tyrosine. So you cannot make the melanin of the eye. And even if 1 is working but 2 is not working, you're not gonna get the melanin. You need both to be active. Epigenetic effects is when gene expression is altered without changes in the base sequence. These are called as epigenetic inheritance. And cancer is when tumors, you see, there's a different types of tumor. There's invasive tumor and there is a malignant tumor. What's the difference between invasive and malignant?
Malignant is isn't that when it's not where the cancerous cells aren't active?
Malignant and invasive. Both of them are cancer. Invasive cancer, it means that the cancer cell is invading locally.
Right.
So you have God forbid, bone cancer or liver cancer staying where it is. Malignant is when it branches off and goes in the bloodstream and swim very far away from the bone to the brain, from the brain to the lung. So that's where you see. That's the tumor here in blue. So the malignant cell goes into the blood vasculature and travels until it goes to a far distant site. That's malignant metastasis, called metastasis. Invasion is when you're infiltrating nearby only, locally.
Yeah.
Okay. Alrighty, carcinogens. So that shows you the kidney tumor. That's an example of the Wilms tumor, and Okay, and that will be all for today.